everybody. My name is Jessica Anderson. I'm a senior in the college studying biology. Thank you for visiting my poster on the role of BAP1 in the enteric nervous system. So the first question you probably have is, what is the enteric nervous system? And the enteric nervous system, or ENS, is the extensive network of neural crest-derived neurons and glia found in the bowel, controlling bowel motility. The second question you probably have is, what is BAP1? And BAP1 is an epigenetic modifier that's involved in regulating gene expression. So you're probably now questioning, why are we studying these two things together? And we're studying them together because previously BAP1 has been linked to decreased expression of genes involved in neural crest migration. So what this means is that BAP1 has been linked to decreased expression of genes involved in the migration and colonization of the bowel by the ENS. And in order to answer this question of the role of BAP1 in the ENS, we're primarily using a mouse model in which BAP1 is conditionally deleted in tyrosinase positive neurons, which is only a subset of the adult ENS. In these mice, we see that they are significantly smaller than their litter mates, and they do not gain weight at the same rate as their litter mates. They die prematurely, and when they die, we see they have something called megacolon, where there's fecal accumulation in the colon. So what we normally see this associated with clinically is a disease called Hirschsprung disease, and that occurs when the ENS does not fully colonize the bowel so that the distal colon does not contain any neurons. So in order to determine if that's something that's happening in our mice, we knocked out BAP1 in about 90 to 100% of enteric nervous system cells, so our neurons and our glia, and that's using the SWINT1 promoter. And then we looked at the neuron density in each bowel region, and we find that there's no significant difference in either the submucosal or the myenteric plexus. And these are just the two nerve plexi of the bowel. So by showing that there is no difference in the total neuron density in the distal colon of these mice, where BAP1 is deleted from all neurons by birth, we're showing that there's no migratory or colonization defect in these mice. However, in our conditional deletion model, we see a disproportionate loss of TCR-positive neurons by P15. So total neuron density is decreased in both the myenteric and the submucosal plexus. And then we show that these neurons being lost are tyrosinase positive, so the ones that are missing BAP1, because we see a decrease in the percentage of the bowel that is TCR positive in our knockout mice. And as a rule of thumb for all these figure panels, our lighter scale bar, our lighter bar is our knockout mice, and our darker bar is our controls. So unsurprisingly, given we know that these mice are experiencing neuronal loss later in life, we find that at this age of P15, there is slower small bowel transit. So the slope here, blue, is our knockout mice, and we see that the transit is lagging behind our hets and wild types. We also see that these mice have fewer colonic motor complexes, which is just to say that their colonic motility is also slower. So in vivo and in vitro assays show slower motility in the small and large bowel of TCR-positive BAP1 mice, which reflects that there is loss of neurons across these bowel regions. So the next thing that we would like to determine in our work is why these neurons are lost, and to do so, we aim to characterize expression abnormalities in neuronal populations by collecting single-cell RNA-seq data from P5 TCR-positive BAP1 flox mice. So if you have any questions after reading through this poster on your own, I'm happy to answer them via email. Thank you so much for listening to me, and enjoy the rest of the Research Expo.